Hello everybody and welcome back to another episode of Catgirl Analysis. Today I'm going to be sharing my notes on how to create catgirls. This is going to be part of a 3-part series. This is merely a summary, some notes that I've collected in my research, and I'm going to expand on it much more later in the series. Okay, now without further ado, let's get started. CRISPR-Cas9 genetic modification, also known as clustered regularly interspaced short palindromic repeats. So through my research, I discovered something known as CRISPR, a gene editing technique. And the Cas9 refers to the enzyme in which we use to split DNA at targeted locations, which allows us to extract and insert a piece of another DNA. And combining these two methods into one actually allows us to create precise and targeted modifications to the DNA of an organism. But there's only one problem. We actually need something to tell our Cas9 enzyme which parts of DNA to bind to, which is exactly why we're going to be looking at guiding RNA. So, RNA is half of DNA, right? Naturally, it's going to search for a part of DNA to deliver itself to. So we can use RNA to bind with our Cas9 enzyme and direct it to the exact location in a genome where we need to cut. And we can use the same method to guide our insertion genes into a missing piece. You see where I'm going with this? We can essentially modify the human genome by inserting the cat genome and creating a catgirl, ideally. Identifying genes in cats to insert into the human. Cat ears. Modifying the FGF3 and FGF10. So the F stands for fibroblast, and this is essential in the development of the outer ear structure. Now, if we can modify this to insert that of a cat, we could hypothetically result in the development of cat ears. But we'd also need to modify the SOX10 and MITF genes, which are the genes that are responsible for the formation of the inner ear and the pigmentation. Now, a cat tail. So the Hox gene family plays a role in the body patterning and limb development. Adjustments to these genes could facilitate the extension of the vertebral column to form a tail. So specifically, the TBX4 and 5 can actually aid us in developing a growing and a functional tail that can actually move. Now, tails in humans is really tricky because we're not actually designed to have them despite having a tailbone, so it would be interesting to see how this plays out, which we'll look at further into the research. So for agility, let's look at the COL5A1 and the 5A2 genes. These are associated with connective tissue. Now, if we modified these to match a cat's, it would result in the enhanced flexibility and increased agility, and it would also most likely contribute to that the feline startle reaction, if you know what I'm talking about. So we get that enhanced reflexes. For senses, I'd say this is optional, but the OPN4, aka the melanopsin, is responsible for light sensitivity and the circadian rhythms. So if we enhance these genes, we should improve low light vision, but we'd actually have to be careful to not completely reverse the light sensitivity in our catgirl, which would basically mean, oh, they hate light now. Creating the first embryos and synthetic womb growth. Okay. So we know how to create them, but the problem is actually growing them. So obviously they're going to have to be grown in a lab. Now that we've outlined our desired traits, used the CRISPR-Cas9 method to extract such traits and combine them with the human genome, we've technically created the first catgirl DNA. So now it's time to actually create them. So we can do in vitro fertilization. This is the best way to synthesize embryos at the blastocyst stage. This is basically when all that exists is a cluster of cells, and it's not even really an organism yet, it's just a cluster of cells. But we can actually synthesize that if we get it at this stage. We insert our catgirl DNA and we begin the growth. Alternatively, we could also put them in the artificial womb. Well, actually, we would need to do this for controlled development and monitoring. This isn't too hard because the creation of an artificial womb is apparently pretty easy. I didn't know that we can do this, but yeah, we can kind of just like put them in a machine that just mimics that. Obviously, we wouldn't be, forcing the growth of an experimental creature onto an actual human. However, this should hypothetically result in the successful growth of an infant catgirl. So let's put it all together. This is how I plan to create catgirls using the methods described. As stated, my process begins with identifying and selecting specific genes responsible for the traits of cats, which we will integrate into the human genome. Key genes will be the SOX10, MITIF for ears. The TBX4 to 5 plus the HOX gene family would extend the vertebrae and form a tail. Then we're going to add melanopsin for the enhanced senses such as sight and hearing. The next step is to grow the embryos for our synthetic catgirls. We're going to do this through vitro fertilization, where a human egg is going to be genetically modified at the blastocyst stage by inserting the catgirl DNA that we created. At this point, the embryo is only just a cluster of cells, so it's most susceptible to the genetic manipulation. Now, our edited embryos are then going to be put inside an artificial womb where we're going to monitor and their development and their growth through testing and imaging. And at birth, we're going to immediately begin evaluating our catgirls and training them as we would with a human. They're going to be kept in a lab environment, as we would need to help them undergo various physical therapy because they will be the very first of their kind. Sensory training and medical monitoring will also be essential to successful training of catgirls. It's likely that some complications will develop with our first batch of catgirls. Now, worst case scenario, we'll just have to dispatch them. Here are my works cited in 12-point Times New Roman MLA 9th edition format, and as always, thank you for watching.